For those of you that joined us a little late, welcome. This is track 5, nano devices and nano sensors. I'm the session chair. I'm joined by Shumei Wang, who's session co-chair. I think in the interest of time, I'll just introduce myself. And I'm the second speaker. And today I'm going to be talking about the functionalization of diatom nanostructures for device applications. My co-authors on this work are two of my recently graduated PhD students I'm very proud of. One is Clayton Jeffries. He's now assistant professor at Catholic University in Belgium. And second is Debbie Gale, and she's a research scientist with Life Technologies. I'd also like to acknowledge the support of the research to the National Science Foundation and through the Oregon Nanoscience and Microtechnologies Institute. Today I'm going to be talking about microorganisms which actually produce semiconductor nanomaterials. And the specific sort of organism that we work with is called a diatom, and it's a photosynthetic algae that possesses a micro-scale silica-based shell. So here's a picture of the living organism right here. The green material is the chlorophyll contained in the chloroplast within the organism. And when you see this outside feature here, it's actually composed of glass, so it's transparent under a light microscope. But if you just remove those cellular materials, you expose this intricate nanostructure. And it's hierarchical. If you look at the structure at increasingly higher magnifications, you see at the submicron scale this very ordered lattice that resembles a photonic crystal array. And actually embedded within this submicron core array is a series of fine structures and even finer nanostructures here embedded with them at scale features below 10 nanometers. In addition to the ellipsoidal organism I just showed you, they also have centric shaped organisms with a radial symmetry. And one feature all of these organisms possess is that they have actually two shells, an upper shell and a lower shell, and then all of the cellular material is contained within them. So about the past 10 years in my laboratory, we've been exploiting these organisms for nanotechnology applications. And we do this through a three-step process that involves bioprocess, or a four-step process that involves bioprocess engineering, material science, and device fabrication. So since the organism is essentially doing all the biofabrication work, the first thing that we do is develop systems to basically cultivate identical micro devices, if you will, on a mass scale through bioreactor cultivation. Once we have that material in hand, then we actually develop a thin film from this material through a variety of techniques which I won't go here. And then once we have this thin film, then we can functionalize it either with biological molecules or with additional semiconductor nanophases. And then finally, we take this functionalized diatom-based thin film, and we actually make devices out of it. And the three devices that we've considered to date include a diatom-based immunocomplex biosensor, a diatom germanium oxide doped electroluminescent emitter, and a diatom titanium dioxide based dye-sensitized solar cell. And today I'm going to be talking about two of these applications, the biosensor and the electroluminescent emitter. In terms of platform level technology, we have two basic approaches. The first is biological insertion of metal oxides into the semiconductor nanophases, actually while the cell is living. And then in the second approach, we take the isolated diatom biosilica and we actually chemically or biologically functionalize it with additional moieties to impart unique function and properties to the material. So I'm just going to start by overviewing the biological approach. using an example of a germanium oxide biologically doped semiconductor material that serves as an electroluminescent emitter. So this is where the biology part comes in. If you look here, here's the cell suspension culture of those microscale organisms I showed you earlier. And about 1 cubic centimeter contains over 1 million of these. They're identical. We use a externally illuminated bioreactor to cultivate these organisms because their photosynthesis requires light as an energy source and actually carbon dioxide as a carbon source. But since they have inorganic-based shells, we also do a programmed feeding of dissolved silicon and dissolved germanium by these organisms, and these organisms take up these precursors in the soluble state and then fabricate nanostructured materials from them. So, briefly, if the organism is in a state where it's about ready to divide, normally in the center there's just the cytoplasmic materials. But when the cell is ready to divide, it forms a plane of cell division. And on either side of the plane of cell division, it sets up an organelle that orchestrates the fabrication of the nanostructured metal oxide phase. So this organism can do it against a concentration gradient. It has a silica transporter built into the cell membrane, which essentially pumps in the soluble silicon, or in our case, we also feed it a foreign substrate, soluble germanium. This material is then polymerized into nanoparticles based on an organic template phase within this deposition vesicle. And then this allows you to impart foreign metal oxide nanophases embedded within the silica matrix. So we can control this process by determining when in the cell cycle the organism is most likely to take up a foreign metal, and then controlling the ratio of those two metals and the total amount that we add to design a certain composition and production capacity for making these nanophases. And in this particular experiment here, we added enough silicon so that the cells would undergo just one cell division. So shown here, the organism takes up silicon. That silicon is used to make a second shell for each side so that when the cell divides, a new shell is formed. If we add in germanium concurrently with this silicoid being formed, we can actually make a silica-germanium nanocomposite on one side of the organism. And we can design, like I said, the composition associated with that. Now, what we found is that when we put this material into the diatom shell, we enable the photoluminescent property to the material. The actual native biosilica itself has some level of photoluminescence due to the fact that it is a biogenic material. This biogenic material has silent on groups that impart some photoluminescence to the material. In our particular analysis, we just used a 337 nanometer UV laser to excite the material, and then just looked at the emission from that material. When we impart these germanium oxide defects into the silica nanostructure, we greatly increase the photoluminescence emission capacity of this material, and we also shift the center luminescence into the blue range. So this gives you an illustration of how we can grow up the organism and then control the composition of foreign metal oxides into it. In order to enable this process for integration into devices, one put this ultimately onto a thin film. And the way we do this is actually pretty simple. After we grow up the cells, we just put in, for example, conductive glass that you could use as a substrate for various optoelectronic device fabrication. And the organisms will actually settle onto the glass, and they'll move on the glass here. I don't have a movie to show you like the previous speaker. But they'll actually move and self-assemble into a dense film. And then if you just take that dense film and use, for example, oxygen plasma treatment, you can basically take off all the organic materials, add carbon dioxide, and leave behind the silica shells, in this case on an indium tin oxide conductive glass layer. And then in this particular application here for looking at electroluminescent capability is that we simply used atomic layer deposition to put on a hafnium silicate dielectric, then put on another aluminum back electrode by physical vapor deposition, and then simply just charged up the device under AC current. So we built the emitter biologically, and then we integrated it into a device using pretty basic, pretty standard semiconductor device fabrication technology. So here's an example of the type of emission characteristics you can get from this. So if you just use the silica itself, you don't get any signal, but when you have the germanium, biologically doped germanium diatoms in here, you get a pretty strong focused emission. If you just look at it with the naked eye, it looks kind of blue from electroluminescence charging. But if you look at the full spectrum, you see some interesting things. The first thing that you see is you see a series of near lasing lines in the UV. And then complementary to those are the 2 lambda resonance lines associated with those primary emissions. And I can't go into the details here, but we've done some other photonic crystal calculations that based on the pitch of the pores and the lattice constant is that these are predicted, nominally predicted, outcoupled wavelengths that you get as a result of this nanostructure. And that it's not perfect here since this is a biologically produced organism. It's not producing a single lasing line, but it's showing about 3 primary ones here. So that's an example of one sort of device that we can make. The second one that I'll talk about is a nano-enabled biomolecule sensor that uses the diatom frustral as the sensing and reporting platform. So in detection for antibody antigen binding, you have 3 steps. The first step, you want to challenge the sensor with a mixture containing the target molecule. And then the second step, you bind that target molecule to an antibody on the sensor. So in this case here, here's your mixture with the circles and the diamonds given, and only the diamonds are recognized by the antibody. So it enables you to pick those out of the mixture, and then once you've got that accomplished, you have to report that. And this is where the difficulty comes in. LISA method is commonly used, but that's a labeling required method. You can use optical methods such as SARS, but that requires some complicated instrument that you have to have. You can also do mass measurement, for example, with a cantilever, or you can do electrical measurement. But all those require devices associated with them. What we wanted to have was a system for standoff detection. And the way we did that was using this native, using this intrinsic property of photoluminescence associated with these materials that we could make. But before I show you that, first I just want to overview the chemistry of just how we got the antibody onto the surface. Unlike glass, biogenic silica has a lot of silanol groups that come in it because it's an amorphous material. So it's readily functionalized to have an amine functionalized surface, in this case through amino trifocylsilane. And then with those reactive amine centers, you can use a common biomolecule protein cross-linking reagent. In this case, we used BS3. That provides about a 1 nanometer linker, and then you can couple the antibody directly to it. In this case here, we used a rabbit immunoglobulin G antibody, just a model system. The picture here indicates that we get pretty uniform coverage of the amine groups to prep the system for functionalization. So schematically,